Hi, I'm Lia Kent from Stemgent. Today we're going to show you how to thaw and freeze human embryonic stem cells. So let's get started.
To thaw human embryonic stem cells, a cryovial is removed from liquid nitrogen and quickly thawed in a 37 degrees Celsius water bath. The cell suspension is transferred from the cryovial to a conical tube, diluted with human ES cell culture medium, and centrifuged to form a small cell pellet. The supernatant is then removed, and the cell pellet is resuspended with fresh human ES cell culture medium in the tube before plating on one well of a 6-well cell culture plate previously plated with an irradiated MEF feeder layer. One day prior to thawing human ES cells, plate a feeder layer of irradiated mouse embryonic fibroblasts, or MEFs, in MEF culture medium in one well of a gelatin-coated 6-well tissue culture plate. Incubate the plate overnight at 37 degrees Celsius and 5% carbon dioxide. To ensure an efficient and successful thaw, be sure to have all necessary equipment and reagents ready before removing the human ES cells from liquid nitrogen. Using metal forceps, remove a cryogenic vial of human ES cells from the liquid nitrogen storage tank. Roll the vial between gloved hands for 3 to 5 seconds.